Yes. Okay, I'll repeat that. Hi, everyone. Welcome to another iteration of the Omics workshop, the Omics Club workshop series. Today, we'll be talking about ancestry analysis, and this is coming from Sri Raj. Sri is a new faculty position here at Einstein. She did her postdoc at Cornell, and she has a degrees in biological anthropology and population genetics from Oxford and a PhD in Cambridge. And she's very interested in ancestry in the Bronx to help improve our patients' outcomes. And that will be followed by a vignette from her student, David Yang. He's a third-year PhD student in the labs of Sri and John Greeley. And yeah, stay tuned for good science.
Thanks, Marley, for the introduction, and thank you to Omics Club for having me today. I'm going to give a little vignette on the basics of ancestry. Some of you who've attended the molecular genetics class, some of this material will be familiar, but it's worth it to go over it a little bit before David gives the overview on some of the methodologies that are involved. So what really is population genetics? It's just the study of genetic variation within and among populations. And this genetic variation can be used to reveal information about population history or population-specific variation in diseases, as well as disease risk. And I don't know if you've heard a few of these quotes, but Dobzhansky said that nothing in biology makes sense except in the light of evolution. And Michael Lynch, who's a population geneticist, added to that nothing in evolution makes sense except in the light of population genetics. And of course, all of this goes back to Darwin, right? And his quote, which is, "From so simple a beginning, endless forms, most beautiful and wonderful, have been and are being evolved." So the take home here is that population genetics is really a branch of evolutionary genetics, and it's a way for us to understand our origins and how we will continue to evolve in the future as well. Now, speaking of evolution, our position in the evolutionary tree is relatively recent. Humans overall have lower diversity compared to other species, and this also lends itself to our understanding of differences between human populations, right? We can all marry one another on the planet. We might look a little bit different, but genetically, we're about 99.6% identical. And if you compare what this means in terms of our nucleotide variations, so the number of variants in our genome that differ, we only have, these numbers need to be updated, but it says the average number of variants per individual is about 3 to 4 million. And you can see that there are more when you look at other primate species as well. And another very important concept for population genetics is this Hardy-Weinberg principle. So G.H. Hardy inferred the genotype proportions in a randomly mating population. And that can be described through this equation, the allele frequencies and the genotype frequencies in a diploid population, right? We all have diploid genomes, two chromosomes, and it's just p squared, which is the homozygous dominant genotype frequency, 2pq, which is the heterozygous frequency, and then the homozygous recessive frequency, q squared. The sum of all of those frequencies should equal to 1. And you can also calculate the allele frequencies, or the frequency of p and q, from this equation. This is a very simple equation, but it's incredibly powerful with population genetics because you can estimate the amount of diversity at the nucleotide level based on this equation. And they make certain assumptions, right? Which is that we have an infinite population size, there's no mutation, selection, migration happening, and that random mating is also happening. And although this seems kind of unrealistic, more or less, most variants in the human genome follow Hardy-Weinberg equilibrium. So then what can lead to departures in Hardy-Weinberg? It's all violations of those assumptions that I just mentioned. So mutation, right? Mutation, migration, gene flow, and I'll go through some of these in the course of this small introduction. Natural selection, population structure, non-random mating, and finally genetic drift. So I'm going to skip the mutation part because we're focusing on population structure for this vignette. So let's look at migration, which is gene flow from one population to another. Humans are among the few species on Earth to occupy every habitable environment on Earth. And it's incredibly rare, especially considering the vertebrate lineage of organisms. And you can see from these maps, these are kind of, the stories that we originated in Africa from about 350,000 years ago, the modern human lineage. And from about 50 to 70,000 years ago, we emerged out of Africa and spread throughout the rest of the planet. And this map here on the, I guess your left-hand side shows how complex some of these migrations occurred, both within Africa and also outside of Africa. And migration also has a very important signature at the granular level. So this is a very famous example of gene flow that happened in Asia around the time of Genghis Khan. So what we've seen through the invasion of, through the historical knowledge of the invasion of Genghis Khan throughout Southeast Asia, Southeast Asia and Central Asian steppes, is that there's a Y chromosome lineage in 8% of Central Asia that originated in Mongolia 1,000 years ago. These are all attributed to his invasion. And this pattern of variation persists in today's society. And now let's talk about population structure non-random mating. So there are two kinds of mating that we can think about. There's positive assortative mating. So you're kind of mating with individuals that share the same type of phenotype. And then there's negative assortative mating, where there are different phenotypes, and that also maintains genetic variation, assuming that these phenotypic differences are caused by something that's genetic. And so a part of this non-random mating phenomenon is inbreeding or consanguinity. So when people continue to marry within the population, a substantial portion of human populations globally practice first cousin marriages, for example, to maintain property with their family or cultural ties. And this has genetic consequences, which means that there's more homozygosity and reduced variation within a population. And in fact, inbreeding and consanguinity is incredibly common. So I said it's fairly common, but it's actually about 10% of marriages globally are first cousin marriages. And we'll talk a little bit about how consanguinity can influence genetic variation later on as well. Another important concept, which you'll hear more about from David today, is principal components analysis. So this is not a concept. It's a statistical methodology that's used as a dimensionality reduction method. And so what does this mean for human genetics? Basically, when you have a lot of genetic data, right, we have whole genome sequence information or genotype data across the genome from like now the order is like 1 to 10 million genomic markers. You want to somehow make sense of the variation at this scale. And so principal components analysis has kind of become a very powerful and handy tool to collapse that variation into something that's a way of summarizing that amount of genetic variation. And so what you end up getting is if you plot a scatter plot, for example, and so the principal components you get goes in order of importance to the variation represented by the data. Principal components 1 and 2 are obviously the most important components that, back up. Well, I don't know what just happened. Sorry, guys. Pause, share. Yeah, I think something, I don't know. Just come back up?
Fine.
Okay, great. I don't know, maybe my laptop. Oh, it's a Zoom. Okay, resume phrase. Is this okay?
Yes.
Great, thank you, guys. There's nothing in the way in the Zoom. Okay, better? Okay, great. And so principal components 1 and 2 are the most important components explaining the variation present in the genome when you use this method. And it turns out that if you plot PC1 and 2, the variation that you see approximates genetic ancestry, which means the most important thing that describes all the variation that we see in this room is our ancestry rather than something else like a disease, like genetic disease origins or something under selection or random mutations. It's our ancestry that explains the most genetic variation as approximated by principal components analysis. And this figure here is actually a really interesting one. I encourage you guys to read this paper because it showed when they took individuals where they had all four grandparents from every country in Europe and plotted their genetic variation, well, did principal components analysis, collapsed that genetic variation, and then just plotted it on a scatter plot, these individuals on a scatter plot. Basically, the location of these individuals on the scatter plot of PC1 and PC2 approximated their physical locations in Europe. So you can see, for example, in the purple and the lavender over here, that's Spain and Portugal. And then if you look over to the right of that, you see France, and next to that, Germany, and above that, the Netherlands, Great Britain, Scotland, etc. It's incredibly fascinating. And this guy won a MacArthur Award for finding these results. So PCA can also, although it's a very simple, basic statistical technique, it also can make some very big findings that have really changed how we think about human genetic variation. Okay. And then we're going to talk about genetic drift, right? So this is not anything, genetic drift really talks about stochastic or random processes. So assortative mating, people are actively doing something that's non-random. Same with migration, right? This is not necessarily a random process. People are migrating to certain areas, et cetera. There's something that's involved in that. But there's also stochasticity, completely stochastic processes that are involved in explaining human variation. And you can approximate that. These are simulations of genetic drift. And but there are other population phenomenon that can create genetic diversity stochastically. And one of these examples is a bottleneck. So I explained earlier that humans originated in Africa, and then some subset of individuals from Africa spread throughout the rest of the world. And so this was a bottleneck, right? Because not everybody from Africa is going to the rest of the world. It's some small subset of it. And not only some small subset, but it's a very small diaspora of individuals that then traveled there. And this creates something called a founder effect, which means it constrains the amount of variation that's represented in the larger population. And then whoever follows that is a representation of that subset of diversity. And it also has relationships to population size. So when you have a smaller population, there are larger frequencies, allele frequency fluctuation compared to larger populations. And it also it reduces diversity and heterozygosity overall, this kind of effect. And this one you'll also see later on in my talk. Now, with all of these evolutionary processes in mind, let's back up a little bit and think about the United States, right? So luckily for us, the entirety of the world's diversity is represented in the United States. Everybody came from all over the world to settle here. And in addition to our Native American forebears who were the first and initial inhabitants of this country. And cities like ours, New York City, are incredibly diverse and are really a microcosm of the global diversity that we see. And so, you know, it's said that over 800 languages are spoken in New York City. I keep saying this. I think this is true. And anecdotally, I hear from Paul Meisner at the CTSA that when he visits a single Bronx high school, over 150 languages are represented in a single high school. This is mind-boggling amounts of diversity that we have right here in our backyard in the Bronx. Now here, this is a map which shows that the two major languages spoken here are English and Spanish. But when you look at the second most spoken languages in each neighborhood, a more complex picture emerges. And some of the populations that live in the Bronx may be founder groups, like these individuals that split up from the larger population and came and settled here. And so here, in a place like the Bronx, thinking about ancestry is incredibly important, right? Because everybody here seems to have some kind of an identity based on race. For example, you're Black or you're white or you're Latino. And if you don't have that self-identity, somebody else will ascribe it to you, because that's how most of variation, at least in the health system or census or these kinds of things, are considered at the race or ethnicity level. But it's incredibly subjective, and it's very helpful for social determinants of health, but it can be misleading when we're thinking about biological determinants of health. See, you know, if somebody says they're Black, does that mean they're African? Are they African-American or Afro-Caribbean or Brazilian or Creole? And Latino is the same thing, right? Are this individual Mexican or Cuban or a mix of different Latino groups? Are they indigenous? That also is considered under the Latino umbrella sometimes in this country. Or if you're white, are you Northern European white? Are you white Hispanic? Are you Jewish? Are you Hutterite or Amish or Italian? All of these identities are a little bit subjective when we think about race and ethnicity. So if we think about genetic ancestry, we are thinking, we kind of define it as global and local ancestry. Have any of you done 23andMe or Ancestry.com tests before? Yeah, I see some hands. Okay, a couple of you. So not too many people, but you must be familiar with this concept that people, take some amount of cheek swabs or something and then figure out kind of what your ancestry is. So there's global ancestry, which is the proportion of different different continental ancestries represented in your genome. And that's what you see here on the bottom. I guess this pointer doesn't. Okay. Well, on the bottom, you see some kind of like a plot, right? And the homogeneous population means like they're almost 100% whatever that color ancestry is. So African is purple in this case. Amerindigenous is blue, green, and European is blue. And then if you look at the two-way admixed population, you see that they're a mixture of two of these continental groups. And so every vertical line, so you can't see the vertical lines, but this is basically a bunch of vertical lines that are all squished together with every vertical line representing an individual. So further toward the left-hand side, you see individuals in the two-way admixed plot that are mostly this green amerindigenous ancestry, and all the way further towards the right-hand side of the two-way admixed plot, they're more European with a little bit of amerindigenous ancestry sprinkled in. And then in three-way admix, which is very typical of what you see in Puerto Rican populations, for example, you see three different ancestries that are represented. And so that's global ancestry, with every line being an individual. But then you might have two individuals that have the same global ancestry proportions, but where this ancestry is located in the genome differs. So that's what this concept is over there on the far right-hand side, the local ancestry plot. So you can see that these two men, or I don't know, stick figures, have the same proportion of global ancestry, but next to them, you can see that on the first chromosome, there's a big chunk of purple in the first individual, and the purple is located somewhere else in the second individual. So you kind of need an understanding of both to see where what variants come from what background and where they originate. And we'll hear a little bit about this from David. Now, I took this slide from my postdoc, Mariko, whose photo you'll see later. And local genomic ancestry can explain complex ancestral backgrounds like the Genghis Khan example I gave earlier. So I showed you those, those two chromosomes earlier, right? Now we can expand that out to a karyogram. And this is something very similar to what you might get if you're a very admixed individual from a 23andMe readout, for example. And you can see that these chromosomes are a mosaic of all these different ancestries. And there are methods that we can use to decompose that, but that we're not going to discuss today. We'll talk mostly about the global ancestry and the PCA in today's vignette. And so my lab does a combination of local and global ancestry and a range of data sets and health implications of that, which I won't get into, but I'll show you a little bit of some of our results from the All of Us research program. And if you guys have any questions about this data set, please feel free to come talk to me. I think you might be getting a vignette on this much later on, but it's not on the schedule yet. And so we took all the genome data from the All of Us research program from New York City participants, and we wanted to ask, what is the population structure of New York City individuals? And here you can see this principal components analysis plot of the individuals. So in the colored dots represent reference genomes. And you're going to hear more about this from David as well. But there are things like the 1,000 Genomes Project or the HapMap project. Ah, man, again. Sorry. Okay. This is gonna, okay. Or the HapMap project, and they basically provide reference genomes so that when you sequence an individual and you plot them on a PC, you kind of want to see roughly what the ancestry is of your participant. And so the ones in colors come from these 3 reference genome panels that I have listed at the bottom. And so you can see that this kind of triangle plot, which is very familiar to those of us who've looked at PC plots before. So out in the top corner is the Europe cluster. It's a very tight little blue group. Africa, you can see, is orthogonal to that group, and it's kind of like a little more spread out pink line. And then you see East Asia, South Asia, and Middle East somewhere in there. And the gray dots in this figure represent our data from the All of Us project. And you can see kind of how it falls on this ancestry continuum, right? They're kind of somewhere between Africa and the Middle East, and somewhere in the, not in Middle East, Africa and Europe, and somewhere in the middle in the Americas, and then there are a few spots out by the Asia lines as well. And if you look at the admixture plots, so if we take each of these individuals and calculate the proportion of ancestry from each of these continental groups, you can see that in the Bronx, we have more individuals that are of this pink ancestry, which is Africa. In Manhattan, we see individuals of more European ancestry. And then in Queens and in Brooklyn, we see a huge chunk of Asian individuals as well. So these are the two representations that David will hopefully be able to walk you through by the end of today's vignette. But I'm going to go into a little more detail just to show you that this is not the be all and end all, right? This is a starting point. And to receive high resolution to New York City populations, we also can do something called identity by descent to look at shared haplotypes between pairs of individuals. And we can get even finer scale structure by identifying networks of individuals that share a lot of DNA. So I talked about the example of consanguinity, so individuals that tend to marry within the population. This is a really great method for identifying these consanguineous groups. And we did that, and using a certain haplotype lengths, so haplotypes are the non-random combination of variants next to each other on a chromosome, we were able to distinguish between different Latino groups, for example, like Puerto Rican versus Dominican versus Mexican, Central South American, and European groups as well. So the biggest differentiator was non-Ashkenazi Jewish versus Ashkenazi Jewish in our data, and Italian, Albanian, South European, and these are all New York City populations. And you can even plot them on the PC, right? So each of those big splotches you saw, the little like circles you saw in this last plot, you can visualize them on a principal components plot as well. Now here the colors are reversed. The gray represents the reference data set, and the colors now represent our data from the All of Us project and from the All of Us, All of Us research program. And so you can see how the different colors reflect the different ancestral compositions of the population. So Dominicans down here in yellow and Puerto Ricans on the upper right-hand side look kind of similar because they're both admixed Latino populations. But then you can see that we have another group like the Central South Americans who are right next to the Puerto Ricans. So these are Mexicans, Colombians, some other Central American populations, and they have a very different axis compared to the Puerto Rican group. And this shows you why ancestry is very important at especially understanding genetic variation, right? Because with race, they'd both be lumped into the same category. But when you look at genetic ancestry, they look very distinct. And so we also plotted the strength of this consanguinity, or the strength of this sharing of segments across each of the boroughs for our populations. And so the strength of sharing, so this is not the number of individuals, it's the strength of sharing of segments. And we identified this big maroon bar, group 9, the Garifuna. The label says South Caribbean African ancestry, so I apologize for the inconsistency in labeling. But essentially, this is the group of people that originated, they're called the Garifuna. Now, who are they? Now, these individuals are descendants of a group of enslaved people from West Africa. They ended up shipwrecking on the Caribbean island of St. Vincent in 1635, and they were never re-enslaved. And the Garifuna are descendants of those individuals who survived the shipwreck and the indigenous island Carib and Arawak indigenous people from the island of St. Vincent. And they kind of stayed on that island and eventually were exiled to coastal South and Central America, where they still reside today. They don't live in St. Vincent anymore. But what's really cool is that they continue to maintain the Garifuna language. So they're, I think, one of the only surviving populations that still speak Arawak and indigenous Carib languages. And there are about 200 to 300,000 that live in the South Bronx. So these are not just small populations sometimes. They're massive numbers of individuals that we only identify through genetic ancestry. Our health system doesn't know anything about them, even though 300,000 of these patients go through Montefiore Health System. And so I'm also going to show you a little bit about the local ancestry of variants. So we identified a few variants that are enriched in this population that are annotated to be pathogenic. And if you look at the segment of DNA where those variants are located in Garifuna, one of these is located on a portion of the genome that comes from the indigenous ancestors. And then a couple of these variants come from the African ancestry part of their genomes. And what's really cool is that you can even trace this back to the places of origin. So these are examples of a bunch of other variants that I haven't talked about here. But you can see that although it's more frequent, like I talked about the variant in the Garifuna, this DUOX2 gene, right? And you can see that it's of higher frequency in the Garifuna, but it's still present in West African populations where they originated from. So there's an enrichment. This is that founder effect I was talking to you about, right? When you get a bottleneck, some portion of the variation settles into a new area and the frequency completely changes from the parent population. In this case, the frequency increased in the Garifuna, but it's still present in the parent population as well, just at a much lower frequency. And so this brings us to population stratification, because some of us in this room might also use genetic variation to look for disease association. Many of you might have heard of a genome-wide association study, right? That's how they identify a lot of these variants that are associated with the disease. They take a group of cases that are enriched for a trait and a group of controls, and they look for statistical significant enrichment of genetic variants. And that's how they identified a lot of these variants associated with this, especially common complex diseases. It's especially powerful for common genetic variants. But the thing that you need to be careful of, oh, I forgot I had the slide. And that's how GWAS is used to link a genotype to a phenotype. And here you can see that in cases, like if it's associated, it might be a very high frequency in cases compared to controls. Now, it's also you have to be very careful when you construct a GWAS study that your cases and your controls come from the same population. Now, can anybody, if you're up for it, explain why that could be the case? Why would you need cases and controls to come from the same population when you do a GWAS? Okay, I gave it about 5 seconds. I'll explain it. So the reason is, remember I showed you earlier that ancestry really is the most important differentiator when we try to explain genetic variation among populations. So what you can get if you have cases from one population and controls from another is that you're seeing an allele frequency difference, but it's not due to the association with the trait. It's just because they have an ancestral difference in allele frequency. And that could be due to drift, it could be due to population structure, it could be due to whatever. But it's not because that variant is associated with the disease. So that's why it's really important to have the cases and controls from the same population. And so same population is also a little bit subjective, right? Because maybe your cohort is all white people, or it could be all white British individuals. And so we kind of try and approximate it using the global ancestry parameters. Like, okay, they're all seem to be continental Europe or something, but we also, in our logistic regression models, correct for principal components, say the first 10 principal components, to really make sure that ancestry is not contributing to these allele frequency differences that you see between cases and controls. You really want to minimize the influence of ancestry, so you're really just pulling out the genetic associations with disease. And so, I guess you can just move this up a little. And so, David will be bringing up an example from asthma from some work that he and Anthony have been doing in my group. Anthony's not my PhD student, he's giving next week's vignette, but he did rotate with me. And so this is a combination of work that both of them have been doing with me. And so the question really was, what is the global ancestry of a functional variant that Deepa Rastogi, in collaboration with Masako Suzuki and John Greeley, identified to be associated with asthma in obese Bronx children? And basically, Deepa gained a lot of, not publicity is not the right word, but she made a really big finding that Obese-associated childhood asthma is a different pathway from asthma in children who are not obese. And this was one of the variants that mediated it. But of course, she had a Bronx children population. Nobody knows what their ancestry is. And so we wanted to ask what the ancestry was of the local ancestry was of those genetic variants. And Anthony and David worked on this, and I don't want to spoil the punchline from them, so I'll let David share what they found. And yeah, I'm pretty much done. I just want to share lastly that it's really important to collaborate with community leaders when you talk about ancestry, because it can be a very sensitive subject. And these are human beings, right? We can talk about population structure and butterflies or plants and rice, and humans should be treated the same way from a scientific perspective. But at the end of the day, they're actual people hold a lot of meaning when they think about ancestry. So it's really important that we be careful when we do ancestry analysis to make sure it makes sense, to make sure the community is happy with it, and to make sure that we're being sensitive and avoiding stigmatization as much as we can. And that's it for me, you guys. Thank you. Oh, just want to continue.
So now we're gonna walk we're gonna do the vignette, and David is trying to show you how to set it up first. So we're gonna start by going to GitHub, running the repository, and then opening the cluster that Einstein that EU, so we can work on it as we go, you know.
We're starting the vignette, guys, so please get that opened up now. Okay, guys, we're gonna get started with the vignette now. So if you get a chance, please open that up.
All right, I'm unmuted.
You're on Zoom?
Yep, everyone should be able to see everything, including my dog.
Start from, so if start from here. You want me to do this one?
Yeah.
Okay.
So,
Okay, I will do this part first. So if you don't, do you guys know how to get here? So this is here, this page, cluster.einsteinmed.edu. So if you can go there, you have to log in with your Einstein credentials. Let me know, somebody. Give me thumbs up so I know that you guys are on that page. Okay, so
yeah,
thumbs
down would be helpful.
all right. So if you've never been here, right? The first thing that you're gonna do is make sure that this button here on the bottom is turned on, and you have this green light. So you see how David doesn't have it on. So you're going to, if it's not on, you're going to turn it on. And make sure it says active. Yeah, so once you're here and in the login in the first screen, right, you're gonna make sure that this button here, this is turned on, and you know it's on because it's going to say active number 1. So right now in David's screen, it says requested. So you just give it like a few minutes. Yeah. In the meantime, so
I never get this to go green. I've never got it.
but you get it to work.
Yeah, I get it.
Okay.
I know that's what they want.
So do people have like buttons green? Okay. So since if you never been here, you might not have these things over here, these apps over here. Or what we want to do is to call Jupyter Notebooks from here. So you're gonna go to your name, you're gonna hover over your name, you're gonna go to Marketplace. And in Marketplace here, you're gonna see a few apps. And today we're gonna be using Jupyter Notebooks or Jupyter Lab, you know? So you click on that, let's say here, click on Jupyter, and you do use latest version. And you click on that, and that should appear in your home screen, So if you go back to home, you should see it here on top. Are people following? There's a few things on this. Okay. So once you have that, you're gonna click on Jupyter, and then this screen is this. So this is when the workflow will start. So you're gonna click on Jupyter, and a screen will show up eventually. Or Jupyter Lab, whichever works for you. Let's see Jupyter Lab.
Oh, no, errors.
Should just mention that if people should have done this before.
Yeah.
So if they are not able to get it working, there's like a known issue with like too many people using it.
Yeah.
Just
I think
They
we're
can just
running
use
into that
HTML.
issue.
Right. So if you cannot get this to work, in the GitHub there is here, these 2 files here. One of them is empty without the output, and the other one is with the output. So you can just download those and follow along what David will tell you.
I think it's all messed up right now. Get the users. I might have to.
You see, let me see if I can get it through my computer, but
Okay.
continue.
Yeah, I think I might. I also can go from my own HPC screen here. I could do this as well and share it with everyone. Share. Okay, I'm gonna switch to my terminal window here, which has the vignette. The only difference is I'm running it through the command line on VS Code. And what's it called? But this should appear the same whether or not you're on the HTML version or you've accessed it through the Git. But I think ideally what we were trying to show was how to clone this Git and open the notebook and then proceed from there.
I would recommend downloading the HTML file. It
Okay.
has all the images and stuff rendered.
Yeah, let's do that then.
Let me just make a quick announcement. So it's kind of a known issue right now. If too many people are using like the cluster, we have like a whole big party in here. So let's all celebrate. But there is kind of like a known limitation with too many people using it. So it might not work at one time. So apologies for the technical issues. So we will just work along with the .html files. So try to download those from the Vignette website and we'll follow along with that if you can't get the cluster working.
Yeah, not found.
You're having trouble downloading it?
Yeah, hold on.
So
Did I just do that?
yeah, if you just click on that. Okay. And
There
then,
should be like a, there
yeah,
it is.
just press that.
Perfect.
Yeah.
Okay.
And then you can just open that up. Make sure you're sharing to Zoom as well.
Yeah, they should be able to see that. Can you see it on Zoom?
Yeah,
Yeah.
it looks like it.
Okay, sweet.
Okay. So, but why don't we just give like a quick minute to allow people to open the .html files, and then we can continue?
Mhm.
Okay. Should we take a break, like a quick, like a 2-minute break or 5-minute break or something, if people want to?
Yeah.
Go to the restroom? Okay. Okay, we'll talk louder. So yeah, we'll take a 5-minute break at this point, so people can, grab a sandwich or run to the restroom. And also try to get the .html file open. And so on Zoom, if you are not able to use the HPC, please open up the .html file. So yeah, we'll come back in 5 minutes. Thank you. All right.
Cluster.
So nice one. Yeah, by the way, your vignette looks really good.
Thanks. Yeah, I mean,
Sometimes.
see how this goes. It's gonna be interesting. No cluster usage.
Yeah, unfortunately, we've been working with HTML people. Oh, sorry, with HPC people to
I never use
get
cluster.
it working. Yeah.
I've never used
It's
it.
meant to make it easier to show plots,
Right.
and also to use the pre-installed software. But they told us that maybe
Got it.
too many people will use it. So.
Thank you, Shane.
Oh, you did? Oh, perfect. We will return at, I guess, 12:58.
The magic of the code executing will be gone. Good. Let's
It's
see.
unfortunate.
View resume. Full screen.
Oh, are you on that? Lock it up, lock it up.
Hmm? I can. I have everything loaded there, but I guess we're just gonna for keep it homogeneous. But yeah, it should be interesting. They don't get to see the beautiful what happens when you press Control Enter and you get to see the output spit out.
Yeah.
We're just
That's okay. At least they
fine.
have code.
I mean, I could demonstrate it, but it's fine. It's not that big a deal.
What's actually in the content bar? I thought there was a message saying you have to figure out this, you have to make sure that the content environment is.
It has all the software installed.
Is it like a list of like module event, module load? Is it just like?
It's sort of like that. Yeah, it's sort of like that. It's instead of using module, you just use the word conda. It's kind of like that in the virtual environment. So it's basically like a little folder that has the path pre-configured. We thought that was going to be the hard part, getting all the packages installed, but actually it turned out that was pretty easy. And just getting people on the cluster turning out to be the hard part, but it's unfortunate. All right, got a sandwich or something like that?
Oh, I have. I'm good. Thank you. I could just.
Yes.
But this is not the HTML version. It's fine. It's like I could share this, right?
display, that's not bad.
Yeah.
Make sure the pictures are working. We did check on the HTML, but we haven't.
Mhm.
Looked at that, so if you just go down the second picture.
Yeah. The first pictures, and I think I removed it.
Stick with the HTML, but it's up to you. You
I'll
can.
just use the HTML.
You're right.
Yeah, because that's what's been sent out, and it's fine. It's just like typos, like a typo or two. It's a
Oh,
picture.
gotcha.
Yeah,
That's a question.
it's all good. There.
1250. So. Again, thank you very much for your work. Yes, really appreciate it.
Let's see how it goes before I get the thanks. I hope
Okay, so
not
it's
to sleep.
12:58. Do you
Yeah.
want to get started?
Yeah, let's be on it.
So let me make sure.
Yeah.
Okay. Are you unmuted here? Okay.
I should be.
So hello, everyone. It is now 12:58, so we are going to go ahead and get started with the vignette portion of this session. So I will hand it over to David. And again, thank you to David for his time today.
All right. So good afternoon, everyone. Beautiful day out. Thanks for coming in person for those that are here, and thank you to those that are attending through Zoom. I'm David, and I'm going to be going through this ancestry analysis tutorial that I've written up. There might be some typos here and there. We have an updated version that's actually on the GitHub, but we'll just work with this completed HTML file for now. And any sort of difficulty and confusion that you might experience while configuring the setup is perfectly in the realm of doing any sort of computational task. So hopefully, though, today that won't be an issue because, well, we have a completed Jupyter notebook here with all of our analysis and results. So jumping right in, I don't know, I think Shree's background slides have kind of discussed a lot of the background information as to why we do a PCA and how we apply this to, say, conduct a genome-wide association study. And all throughout this vignette, you're going to see text that's highlighted blue. Those are just hyperlinks to various resources that you can click on while we go through this, more on your own time when you revisit this vignette. But before we begin, I just want to give a brief outline to keep us oriented, because this analysis can be quite tedious and involves multiple data sets and then merging these two data sets together, running quality control and whatnot. And so in this first portion that I'll be going over today, we're going to learn how to apply some common methods that are used to take data on human genetic variants, in this case in the form of single nucleotide polymorphisms, and prepare these data so that they're suited for population structure analysis. On the left, within this figure, this graphical abstract, you'll see that there are two starting data sets. In pink, we have the HapMap samples. That refers to a simulated data set of individuals from the HapMap or haplotype map project data set. And on the right is the 1,000 genomes reference panel. And I'll get into more of what these data sets actually are, but essentially, for the sake of simplicity, these are two different data sets comprising of SNP data that was generated using microarrays to actually get that data out. And so first, what we'll do is we're going to start on the left-hand side and prepare the HapMap sample data, and then jump over and prepare the 1,000 genomes data set. By prepare, I mean run several quality control steps, and then we're going to merge these two. And the reason we're doing that is so that we can actually run our principal components analysis, and then downstream of that, we'll also go over visualizing this data, as well as the programs that I'll be using along the way. And so I'm going to skip through this background section. And I guess we can jump right into an example where Sri kind of alluded to prior, where population structure can, if not controlled for, can actually lead to spurious associations. And once again, I've included all the appropriate citations here if you want to actually jump into these papers. As you can see, they're quite old, and the one that I'll be focusing here is this Noller et al. From 1988. And I just want to read this quote to get things started. And so, when cases and controls have different allele frequencies attributable to diversity in the background population, unrelated to outcome status, a study is said to have population stratification. And so in this example that I've highlighted here, which looked at a longitudinal study of type 2 diabetes in two Native American tribes, essentially when the authors this was a bit of an exercise that they conducted and published, when they looked at the rate of type 2 diabetes within their study cohort, which comprised of about 5,000 individuals, they observed an inverse association between this GM3, et cetera, haplotype and case status for type 2 diabetes. And this was, so I guess to backtrack, to simplify, individuals who had this haplotype present in their genomes seemed to be at reduced risk for type 2 diabetes. And this was an association seen when not accounting for the genetic ancestry of these individuals. And so failing to do so would lead to a false conclusion that this haplotype might be acting directly to reduce type 2 diabetes risk within this population. However, when they went back and reanalyzed this data using the self-reported ancestry and family records from these individuals, they saw that this association was not seen at all. And why is that the case? If you look at this first bullet point, essentially, this haplotype actually occurs at very low population frequency amongst these Pima and Papago Indians, but occurs at quite a high frequency. Almost 60% of European individuals tend to have it. This is also so compounded by the fact that type 2 diabetes is more prevalent in Native Americans, roughly 40%, compared to Europeans. And so you have a case where you have elevated prevalence of type 2 diabetes, but the absence of this haplotype in Native Americans, which are the group being studied. And then you have a lower risk or lower prevalence of type 2 diabetes in Europeans, but a high frequency of this haplotype. And so when you actually control for the ancestry of the individuals within the type 2 diabetes study, this is what the real finding more is that this haplotype essentially can serve as a marker for admixture between Native Americans and Europeans. And so we'll be going into some of the methods that are commonly used to actually get at this using SNP data. And the program that I'll be teaching today is called PLINK, and it's quite widely used within the field of population genetics, especially for, say, conducting something like a genome-wide association study. And so before we get started with any analysis, and I'm sure this was covered in the prior RNA-seq vignette, there's a series of quality control steps that need to be conducted to ensure that our data is reliable and accurate. And so we're going to be using PLINK, and PLINK uses a set of its own file format, which is a binary file format that has 3 different components that are represented in 3 different file types. So you have the .fam, the .bed, and the .bib. And so anytime we generate a new PLINK file, which is the output of any sort of PLINK-related analyses, we typically will generate these 3 file types. And I don't want to get too into the weeds, but I encourage you to quickly look through. The data themselves are not anything too complicated. The bed file is a bit hairy because it's a binary file format and not easily readable, but the FAM and .bim are essentially just tables that hold either information on the individual within your sample, as well as their sex, and any sort of phenotype coding, that's this P column that you might be interested in. And then the BIM file will hold information about a given genetic variant. In this case, we're interested in SNPs, and report the coordinates for that SNP, as well as the reference and major and minor allele for that given SNP. And I will whiz through this just because it's there for everyone to read, and jump straight into some of the key QC steps that we're going to be going over. And if you want a super well written better written than mine, tutorial or vignette. I adapted this from a pretty widely used tutorial from Andres T. Maurice that can be found here on the skit. And there's an accompanying article linked down here that actually goes into more of the rationale behind why we apply certain quality control filters and why we apply certain thresholds for doing so. So the key QC steps that we'll be covering today, there are 7. We'll go through this sequentially. And once again, all the files, if you go out and want to conduct this analysis on your own, the files need to be structured in a way so that they're either in the same directory or that things are being referred to accordingly. And that's luckily taken care of for this example. So first, we'll start off by working with the HapMap dataset, which is our query dataset. And so this dataset will have all of the SNPs that we're actually interested in that highlight or that denote the genetic variation within our study cohort. And we'll apply these 7 quality control filters as well to our reference dataset. And so first, we'll start off by individual and SNP missingness, and we'll get into exactly what that means. Then we'll identify inconsistencies in the reported versus genetic segments of subjects. Then we'll filter out variants by their minor allele frequency, identify variants that deviate significantly from Hardy-Weinberg equilibrium, calculate rates of heterozygosity, identify related samples, and then identify outliers and filter out those that are not in agreement with our thresholds. And the reason why we do a lot of these steps essentially is to remove samples that might have genotyping inconsistencies or have low quality genotypes as a result of, say, a technical artifact in the actual sequencing of that genetic variant via, in this case, microarrays. And so let's jump right now into the code. And so before, actually, let me just go quickly over how Plink syntax looks like. Here, there's an exclamation point. That's just for how we write things in the Jupyter Notebook. And if you're able to run this on your own, you should be able to execute it just by hitting Control-Enter or the little play button on the Jupyter Notebook. So Plink, to actually call Plink, you and this is why you have to have that Conda environment installed so that you can actually put in a Plink command and have that program run. So Plink, you call upon it by saying Plink, and it takes in an input file data type, and you specify that here. B file represents the binary Plink file format. And then right here is where we actually have our input data, which is this HapMap3 R31. In this case, I'm going to use the frequency flag to just give me a summary of the genotyping frequency and rates within our data set, and then export those results to the results folder in a file called freak, and give it a memory allocation so that the program doesn't crash. Now, I'm going to go into I'm going over this now because we're going to repeatedly see this blob of text essentially, and I don't want your eyes to kind of just gloss over it, or at least know what your eyes should quickly gloss over and what it should actually be looking for. And so just to be thorough right off the jump, this Plink will spit out in its output, kind of call back what kind of flags you used and what kind of thresholds and what kind of quality control metrics and whatnot. It'll report that out within the first lines of code, as well as the version of Plink. It'll then kind of tell you how much memory was used. And this is when, now right below that line is when you actually start getting a sense of what your data set actually has. And so in this case, we have, what is this, 1 million variants, about 1.5 million variants that are loaded from this .bim file. Those variants are coming from 165 people. And you have about a 50-50 split between 80 males and 85 females. And then you have 112 phenotype values loaded from the FAM file. These are simulated just binary trait outcome for the sake of this tutorial. And we're actually not going to be using that. So don't worry too much about the phenotype values. But if you were interested in conducting, say, like a GWAS, this is where you would you'd have your FAM file loaded with the phenotype or trait outcome that you're interested in. This is just computing information. Okay, and here we have more data about the samples themselves, and I have a description right below this that we'll jump into. And you sometimes might get a warning that depending on the severity of it, in this case, we can ignore. And because we called upon this frequency flag or function, we can get a genotyping rate as the output. And so an in-depth description is shown right here. What I want to highlight kind of starts from down here to keep us oriented. And so we have 165 individuals, and it's split between 112 founders and 53 non-founders. In this case, in this data set, the founders are individuals that are considered the starting point of their family trees, their parents. And then the non-founders, essentially the children of these parents. The warning we are going to ignore for today. And another thing that's to take note of is this total genotyping rate. 0.997, et cetera, refers to the fact that we essentially have a really high genotyping rate, almost 100%, which means that for each given SNP in our data set, there's a low degree of missingness, or there is a high proportion of successfully genotyped markers. And so across the individual. So each individual will have some genotyping call at a given site. And it's almost the entire data set, which is great. And then once again, there's just the output. Okay, so that's as much as I'm going to say about walking through Plink's entire output. Now we're just going to jump into what happens when we start running these sort of quality control filters. And so the initial step before we get into any analysis is to investigate missingness per individual, and then look at the missingness per SNP. And then we're going to visualize this data to see essentially how dirty or clean our data is, to put it simply. And so the way I'm going about this, in this case, is by using this missing flag. And what that will report, if we jump right here, it should appear similar to our previous output, where we have the number of founders, the number of samples, et cetera, the total genotyping rate. And what we see in these bottom two lines is where these results are written to. They're formatted in these .imis and .lmis files, and those will have the number of SNPs, or these files will have the missingness per individual. So each individual in this case will be a row within this larger file, text file essentially, and then the columns will refer to they'll report the missingness of they'll report their genotyping rate for all the SNPs, and then the LMIS does the same, but in, I guess, with SNP as the row instead of individual. Okay, so it's weird, I'm not running any code. So we first start off by removing any individuals that have high missingness prior to filtering out any SNPs. And these are accomplished using two separate functions from Plink itself. So we're going to start off kind of as like a first pass to set a threshold of 20% or 0.2. And in this case, no one should actually be filtered out. This is quite a loose threshold, this 20%. And we're just using it to essentially get a feel for the data, and also ensure that there's nothing egregious with the genotyping that was conducted for this study. And so I've kind of already spoiled the punchline, and we're going to gloss over all of this because we're pretty familiar with it now. Here we go. So as you can see, 0 variants are removed due to missing genotype data. We know that this data set is actually quite of good quality. And so we preserve our 1.5 million variants across 165 people. And they're split into cases and controls here. Okay, we're going to apply the same sort of loose threshold to see what, just as a cursory sort of prospecting run here. And instead of using the genome flag, we're going to use mind, which rather than filtering out samples or individuals based on their or SNPs based on their missingness, we're going to run this for individuals themselves. And I'm just going to highlight once again where I think our eyes should focus on. No one is going to be removed. The data set essentially is the same here. And we run these in the beginning just to, once again, ensure that there aren't any, I guess, screaming outliers for poor genotyping quality. Yeah, so on that note, I will stop now and I guess take some questions if anyone has any. I hope this isn't anything too visually painful or confusing for looking at Plink outputs, because we're going to run through these. And nothing exciting has happened yet. Also, we filtered and done 0. But if anyone has any questions, feel free to interject. Yeah. So yeah, that's the unfortunate part of running the just doing it through this HTML. These commands. So for like, right, 165 people, 1.5 million variants. Given the memory that's allotted, it takes about a couple seconds. Yeah. So the longest some of these commands will run within this vignette take about a minute. So it's actually quite fast. I think, yeah, Plink does a good job of doing that. As you can see, you can also multi-thread. So if you have like hundreds of thousands or something like that, you can drastically increase computational performance.
If it do take a lot of time to keep, like the frequencies, that takes a while for it to calculate, because it has to, get all the frequencies and individually, that takes a while. But there's so many things you can do with Plink. So some of the QCs have to straightforward. Relatedness takes a bit of time. A little frequency takes time. Calculated like this takes time. I'm talking hours, depending on the site.
All right, so let's jump right back in and progress through this QC, because we're going to essentially repeat this for our reference panel as well. And so here, what I'm going to do is now filter out SNPs that have a missingness rate of greater than 2%. And that's once again done using this genoflag and specifying the cutoff threshold that we're going to use. And this is a much more stringent threshold compared to our 20%. And this will ensure that we essentially have very high quality genotype calls within our data set. And not that none of this is important, but because we're going to see this so much, I'm just going to quickly highlight where I want to direct everyone's focus to. And so our first exciting result, look at that. 27,454 variants have been removed due to missing genotyping data. And so, can anyone speculate as to why these variants might be filtered out? Why might they have low genotyping data? There's a bunch of reasons. Let's attribute it in this case to just a bad microarray chip or loading of the chip itself. These poor quality variants can be introduced simply because of a technical step within the genotyping procedure. And so we're just filtering those out. But as you can see, most of our variants, still 1.4 million, remain. And that's good, because it gives us a nice starting query data set to work with. And then we're going to repeat this missingness filter to remove individuals that demonstrate high degrees of individual missingness. So you can have a sample, say, out of your lot that, I don't know, might have a contaminated reagent or something like that, that would interfere with the genotyping reaction on a microarray. And so this missingness filter and this genofilter are what we use to kind of get rid of those artifacts before you can conduct any analyses. And here we are. When you run this more stringent threshold, actually everyone remains. So at the individual level, we see that 165 people, which was what we started with in our query data set, still remain and make it through this first round of QC. And so next, we're going to look for individuals that have inconsistencies in their reported versus genetic sex. The way we do that is, once again, through Plink using the check sex function. And the way this works, Plink will go in and look at the amount of homozygosity along the X chromosome to determine the genetic sex of each individual. And then output these results in a file of your choosing, in this case, the sex check file. And we'll use this to determine if we need to remove any individuals that have this discrepancy, as these can also indicate potential errors in the sequencing. And so this should all look familiar. And here, boom, we see that there's one problem detected. So anytime you have an individual that has a discrepancy between their self-reported sex, which kind of rides along in that Plink binary file format in the FAM file, if that's discrepant with what Plink or from Plink's homozygosity check, this will get noted as a problem. And so we see that there is one, in this case, self-reported woman here with the sex discrepancy, and it computes this F statistic, which is 0.99. And so the following commands are going to be used to actually deal with this. Here, this first line, I'm just using to extract the results of that sex check function that I just ran, looking for the individual that has problem next to it, and just putting that in a separate text file, which we then call back to using Plink to edit our input data set, which is this HapMap version 5 data, and essentially generate a new bed file, or this binary Plink format, into a fresh data set. And so all that changes between going from everything we just went through to down here is the removal of one individual. And the reason is, if you're a male, by Plink, when it runs this sex check, the F statistic that gets calculated, which once again corresponds to the X chromosome homozygosity, should have a value of greater than 0.8. And so for females, that should be less than 0.2. And in this case, we have a self-reported female that's at 0.99. And so we're going to exclude this individual from subsequent analyses. And that is achieved by running this exclusion code. Once again, you take your input, you give it a text file that has, in this case, a sample that you'd like to filter out, and then you have Plink remake the entire file set and export it. And this process of directing Plink using a text file is very common, and is something that we're going to be using to further parse our data down the line. And so the fourth step that we're going to be employing here is filtering our data set now by minor allele frequency. And so we're only interested in this case for our PCA analysis for autosomal SNPs and common SNPs that occur at a minor allele frequency, in this case greater than, I think, where is it? I actually don't see. Never mind. We're first going to just pull out our autosomal SNPs. And so I prepared a text file essentially that only contains SNPs that fall within chromosomes 1 through 22. And that's what this awk command is responsible for. And this requires a grasp of basic command line scripting, or adequate knowledge of how to communicate with your favorite ChatGPT or Bard or Gemini to create this text file. But essentially, all we're doing here is taking out, excluding the sex chromosomes from our analysis to only work with SNPs from chromosomes 1 through 22. And when you pass that over, you shrink your data set to, we lose a couple variants essentially, but we still retain about 1.4 million. And now we're going to, before we actually conduct any sort of minor allele frequency filtering, let's visualize the results. Oh no, okay, never mind. does not matter. So here, if you go down, we'll see a plot of the distribution of SNPs and minor allele frequencies. And I don't think the binning for this graph was done correctly, but it still captures what we're trying to show here. And so on the x-axis is MAF, or minor allele frequency. And this refers to the frequency of the minor allele, which ranges in this case from 0 to 0.5 or 50%. And on the y-axis are the number of SNPs that fall within each bucket of minor allele frequency. So as you can see, there's quite a number of SNPs that fall within this range of 0.1 to 0. So there are a lot of low frequency variants that are present within our data set. And we want to look at variants that are in common or that occur at a relatively higher frequency within the population of interest here. And so we're going to actually filter out many of these low frequency variants using SNPs, using Planck's minor allele frequency thresholding function. And so that takes place over here. And so once again, the syntax should look familiar, where you call upon Planck, indicate the type of input file, which is our binary formatted HapMap file. And now we're going to use a cutoff of 0.05 to remove variants that we'll call, I guess, rare in this case, or have a minor allele frequency that occurs at less than 0.05. So we're essentially filtering out everything, this big bar here. And when you run that, we see, I already have the answer here, but you see that about 300,000 variants get filtered out and about 1 million still remain. And you'll notice that we have only 164 individuals because we dropped one from the sex discrepancy analysis. And we're almost there now into actually visualizing this data and running our PCA. But before we do, we're going to jump back into some classical genetics here and look at how our data set, or see if there are any SNPs in our data set that deviate from Hardy-Weinberg equilibrium. And so here, if you use Planck's Hardy flag, you can have Planck calculate the Hardy-Weinberg equilibrium for each SNP. And all I'm doing here then is using the results of that to create a plot that will visualize the distribution of Hardy-Weinberg equilibrium values for our data. I don't think it's actually particularly helpful for the way I plotted these, so I'm going to gloss over them. I think the results themselves, though, are going to be much more descriptive. And so here, the way we go about this is it's a two-step process where we first take our query data set, still working with the latest version of our SNP panel from the HapMap project. We're going to apply a Hardy-Weinberg equilibrium threshold of 10 to the minus 6. And this is adjustable based on how stringent you want, and that you want is dependent upon the analysis question that you have downstream. And then this first pass filtering actually is only applied to the controls in our data set. And then we're going to rerun this filter for the cases using a more stringent filter. And the resulting data set that we get retains about 1 million variants with 164 people. So let's see. And just to hammer through this, we're going to jump into step 6 here and calculate the heterozygosity rate. The reason why we're doing this is to remove individuals or individuals that demonstrate a high rate of heterozygosity. The reason being is if you have a sample that demonstrates an exceedingly large amount of this heterozygosity, could indicate that there's once again, a potential gene typing error. And the cutoff we're going to use for this is 3 standard deviations. So we're going to remove any individuals that have a heterozygosity rate that occurs 3 standard deviations above what is observed in the data set's mean. And so to run this, we're going to once again use this exclusion text file business to remove SNPs that deviate from Hardy-Weinberg equilibrium or violated its assumptions. Here, there's also another thing that's happening down here, this in-depth pairwise 55.2. So what's happening down here, we're telling Plink to look at a specific window of SNPs to look at, in this case, a 50 base pair window, and to slide that window each time 5 base pairs. And then what does that point to? And the point 2 refers to the correlation between those SNPs, and that's given to us by the Hardy-Weinberg equilibrium. Sorry. And so when you run this, it's essentially going to look for do this sliding window approach to wait. This is not correct, I think. So this is not actually calculating heterozygosity rate. Sorry, this is doing what's called LD pruning. Here, what we're doing here in this sliding window approach is sampling across a 50 base pair window, looking for uncorrelated SNPs to essentially prune down our data set. This is probably disjointed from everything I've been discussing prior, but this is critical for the analysis that we're actually interested in, which is PCA. In this case, we're using the sliding window to essentially prune our data set from million variants down to about 100,000. And this will generate a set of SNPs that are essentially unlinked from each other and independent. And these are going to be what's used for PCA analysis downstream. Sorry, this is not what I was anticipating. And then, right, wow. We have heterozygosity outliers down here. So things are disjointed in this, but if you're still following, I have this Python script that I included that would highlight which individuals deviate from Hardy-Weinberg equilibrium by 3 standard deviations. And these are the 2 individuals that we're essentially going to remove due to this higher heterozygosity rate. And so when you run that, we're down to 1 million variants across 162 individuals. Okay. Lastly, and I want to get through this because we'll have some nice visualization to do in just a bit. We're going to identify samples that are relatedness or that are related. And we're doing this to once again, for a similar reason as to why we're doing this sort of LD pruning step. You want to make sure that the SNPs you're analyzing are independent from each other, meaning that they segregate independently. And then 2, you want to also analyze samples that are independent from another sample. You don't want to analyze related samples. And so to do that, we are going to look for what's called cryptic relatedness. Or essentially, we're going to look at, we're going to use Plink to identify regions that, or not regions, individuals that, to quantify the amount of relatedness within our data set between individuals. And we're going to use this King coefficient or kinship-based inference for GWAS that was developed for Plink to estimate this kinship coefficient within our data set. And we're going to be applying a threshold of 0.125 in this case to remove any sort of second degree relatives. So those are half siblings, grandparents, and grandchildren. And that's once simply achieved by, once again, providing your input file and using this king cutoff flag specifying your threshold. And what we should see down here is, here we are, that actually a good number of samples get removed. So we start off with 162, and we identified 52 that are cryptically related. So these 52 that were filtered out are individuals that are related to the second degree. And so at the end of this analysis, what we have are 110 samples. And so the way we do that is, once again, kind of telling Plink, hey, just keep your unrelated samples in, and generate a new file set that has these, only has these 110 samples. Whoo! So congratulations. We've made it here. And essentially, if you would like to conduct your own genetic analyses using SNP data, you can use this code as a bit of a structural backbone and swap out the input data files and adjust the file structure if needed to do this QC. And I strongly encourage, if you do go down this route, to also refer to some of the papers that were highlighted previously in the hyperlinks above. And so I think, should we take a break here? Questions, water? All right. Yeah, we're gonna take a 5-minute break here. Thanks for sticking around. See you in 5.
Any questions in the Zoom? Yeah, the chat there. I didn't see any.
I don't have it. I don't see it.
I'm guessing everyone is fully.
Yeah, I don't know.
Do you know?
I'm like, I've never actually had to talk through each QC stuff.
Yeah, you're doing great.
I'm
It's right
fucking-
there.
Thank God. I think should move fast.
Yeah, it's like it's 7:46, because it's pretty much the same and Sunday.
Yeah.
So it'll be 1:45 when we start, and
Okay.
you know,
Yeah,
theoretically, we told people until 2, but we did get started a bit late today. So you know, it's up to you if you want to go over it. It's still up to you.
I'll go over the the admixture stuff I think it's quick. There's no code running and it's just Tree also gave a bit of a preview of that, and it's the data set that we're using. I just want to get to that part because I think that part's much more interesting.
Got it. When we do hit the 2 p.m. mark, maybe you could just mention that, you know, we started a bit late, so people can feel free to leave if they need to.
Okay.
And just mention that into
For
Zoom.
this part, I feel like you can just this is just pretty much the same thing we did, like you know, you can just work that on your own and keep going.
You're doing well.
It's just so quiet, and I look, everyone's so far over there.
That's why I was like thumbs up, because I feel like I need feedback from people.
It's a cold room.
Not sure what it's doing.
The weather or whatnot, it's fine.
I had a question for you. You talked about the sex stuff, and that's depending on the like zygosity, I guess, of the X chromosome. But do you ever I'm assuming that you could probably see like things like Klinefelter syndrome, you know, where there's like 3,
Oh,
you know, stuff like that. Do patients with that sort of get thrown out, or?
so like Plink will, so there's a big caveat that I haven't mentioned for Plink for this analysis. I've kind of hinted at it. There's a reason why we're only including autosomes, and there's also another reason why we're only including SNPs, and there's a subset of filtering that was done to only include biallelic SNPs. So Plink does not handle like triallelic variants and things like that very well. So it'll crash. The program will essentially freak out and crash.
Oh, interesting.
And
Maybe
so,
it's like a different tool for that, basically.
yeah, and so I think one big caveat to highlight is that, you know, we're using this to gain, you know, we want to run PCA to gain this understanding of like the granularity in our data and look at how the samples group by ancestry, right? But in doing that, you're not actually like we actually throw out a lot of information, right? You downsample from like 1 million down to 100,000 variants, and you're just using the ones that are the most like informative by.
Maybe we can get started.
Yeah.
Thank
Yeah. So I'll actually start with that.
So you just like say hello on the Zoom. And
Yeah.
Thanks so much, man.
All right, Zoom peeps. I think we're going to resume here. And everyone else in the room. So we're going to jump through this next section, which essentially is just a repeat of the QC steps, except applied to the 1,000 genomes data set that I mentioned earlier, which will serve as our reference panel of SNPs. And so we're on the precipice of conducting our first pass at ancestry analysis. And so that'll require our QC'd HapMap samples, which we just walked through. And then also the QC version of our 1,000 genomes reference data, which comprises of SNPs from 629 individuals from different ethnic backgrounds. They'll serve as our sort of reference populations that we plot against to observe, to see the actual genetic ancestry of our samples. And if you want to actually download this data and use it yourself, simply copy and paste that into your terminal and wait a while, and it'll give you this data set. And I highlight that because it's actually quite commonly used in this kind of population genetics studies. One thing to also caveat before we move forward is, you know, this analysis we're doing here is to gain enhanced sort of resolution of our data set and understand the genetic ancestry of our samples. But as you can see, or hopefully it was visible, that a lot of variants actually get quote unquote thrown away for this analysis. We pruned our data set to only take on about 100,000 markers, and we started off with like about 1.5 million. And just to recap, there's two main reasons why we do this. One, like I mentioned before, we want our samples to be independent from each other, and we want the genetic variants that we're analyzing to be independent from each other. And then two, another key point is that we gain a lot of computational efficiency by down selecting our variants and running these quality control filters. And so when we're dealing with these large text files, essentially of a million different rows and columns and whatnot. Anyone who's worked with computational sort of framework knows that that just increases the time at which you can process these kinds of files. And so we're going to apply the same quality control filters here. That it's just all clumped up into one code chunk. And even more so, what you can do is just actually have all of these quality control filters as one line in Plink, and Plink usually does a good job of parsing that out and applying each filter accordingly. Now I say that caveat of usually because something weird happened when I was writing this vignette. Normally I would just have everything in one go, press enter and get a result, as you guys are all aware of, that looks like this. In this case, I actually went line by line, and now I think I go line by line in my own analysis because even though it's more tedious, you can, one, tease apart each result, and then two, Plink can sometimes run into a handling issue where it almost tries to, and this is a bit of a guess, but when I apply all these filters in one go, essentially no variants remain, as I think it tries to find some weird intersection of all these different quality control thresholds. But we're gonna skip over this. And just highlight the main result coming out of our quality control. And so in our reference data set, and this will make more sense as we move down, you see about almost 6 million variants across our 629 people. Once again, it's very high quality genotyping data, which we're thankful for. But yeah, in this case, we start off with our query, or our reference panel has about 6 million variants as opposed to the 1.5 from our previous query data set. And so before we get into the analysis, now that the QC for both the HapMap data and the 1,000 genomes data is complete, what we want to do is to run PCA, but not run PCA individually, just on our samples and our reference. What we're going to do actually is merge these two data sets together. The merging of any genomic data set, as I'm sure some audience members are aware, is actually can be quite a technical headache and hurdle, and they're actually, it's a bit of a weird unresolved thing in the field of genetics that when I first started working with Dr. Raj, that I thought would have been something simple as just pasting two lists together, but that's not the case. And so here I demonstrated one of the ways that you can go about ensuring that the merging takes place correctly. And in this case, when we do this PCA analysis, what we want to do is make sure that both data sets contain the same genetic variants. So what we're going to do in these subsequent steps before we merge is prepare the data one last time to make sure that they indeed do possess the same genetic variants. And so to get there, essentially, all we're doing is having Plink and the computer go through the query data set in this first line and extract the names of all the SNPs within that query data set. And once again, the query data set has been proved down to only 100,000 SNPs. And so in the second line, what I'm doing then here is taking the reference data that has been QC'd, it's about 6 million variants across 629 individuals, and then telling Plink to, hey, only extract the variants that are seen in the HapMap prune data set, and then generate a new bed file that will only have the intersection of those two data sets. And then what I'm going to do as like a double check now is do the same sort of cross-referencing of lists, but in the flip side. So we're going to start with this output of the 1,000 genomes data set, and then extract the SNPs that are present in there, and then use Plink to edit the SNP data set from our query to only have the SNPs that are found in the edited 1,000 genomes data set, and then make a new bed file called hapmap_PCA. And so this is just like there's just a comprehensive way to kind of go back and forth to make sure that your data sets have the same variant sites represented. And as you can see, yeah, we run here this chunk of code, and you see that a new set of files are generated. Now, another key component to take note of is that you want to make sure that both your data sets are also aligned to the same reference genome build. This is a bit of a crude way of doing that, but this is actually super critical whenever you're comparing not just human genome data, but mouse or any species. The alignment of these data to the correct, and in this case, the same reference genome build is absolutely critical for ensuring accuracy and reproducibility in our results. And so here, we see another awk command. Essentially, we're extracting the SNP IDs for our HapMap data set and contrasting that against our 1,000 genomes data set, and making sure that essentially they have the same build, which they should. These were, I think, aligned to HG37. And so once you do that, so once you have ensure that your variants are the same between the two, and everything is aligned to the same data set and formatted correctly up until this point, now you're able to actually go ahead and merge these two data. Oh, wait, sorry, I spoke too soon. What we're going to do is ensure that, right, the reference genome set, you're going to run into these, in this case, these impossible A1 allele assignments for a handful of variants. Once again, the severity of these warnings can vary. And in this case, we're going to ignore these. And by ignore, I mean, I'm going to scroll right over them. Here, just reporting the new allele assignments that were made, the number of variants within our data set, and the number of people. The other key thing to check for is strand flip issues, where you have certain genotypes being reported on the forward strand or the reverse strand, and you want to make sure that everything is consistent so that you don't have these discrepancies for any given genetic variant within your data set. Here, these awk statements, once again, should appear familiar, where we're essentially extracting columns within our data set, within our Plink data, into these temporary files, and then we sort them so that we can essentially see what differences exist between how the variants are being coded within our reference data versus our query data set. We are, when you actually run, look at this text file, you see that there's there's 1,624 differences between these files. And these are potentially due to these strand issues. There's also a ton of these weird impossible A1 allele assignments that might be triggering this. And so to actually resolve these potential SNP flips, what we're going to do is take our differential list here and use this flip command from Plink to actually reverse the way in which these SNPs are being encoded. And so that is done. Sorry. Where is this flip? We don't have a flip. I think, okay, sorry. I was doing this on my own analysis and I spoke too soon. In this case, what we're going to do is simply remove these these problematic variants that have this weird strand flip issue. And yeah, this requires, once again, some computational knowledge of how to use awk and sort commands, but feel free to, the Plink format is pretty ubiquitous. And so even if you use your own BIM file, you can actually copy and paste this and swap out the file names to actually conduct this snip flip check. And so here, what happens when you actually start extracting these discrepant snips, you'll see that there are 84 differences between our query data set and our 1,000 genome reference data set, which actually shouldn't exist since these files were generated or were edited so that they should have the same variance present within those files. But here we see that there's 84 differential sites between these two files. And so rather than doing what I mentioned earlier, where you invoke the snip flip function from Plink, what we're going to do for the sake of this example is just simply remove these problematic sites. And so here, using another set of awk statements, essentially, we're going to supply a text file that has the subset of uncorresponding snips, and then sort them, rename them as a snip for exclusion file, and then supply this snips for inclusion file into Plink. Once again, using this exclude feature where you can tell snip to take your input and only either keep or remove files that or snips that you're interested in. In this case, we're going to remove these and do the same for our 1,000 genomes data set. And so at the end of all this, what we should have is a clean data set for both our HapMap data and our 1,000 genome data. And in this case, because the lines are written on top of each other, the outputs are going to be squished on top. But essentially all we're doing within these like 10 lines of code is removing 42 sites within both of our files. Finally, we can get to the merge. And so up until this point, we've addressed some key weird issues that could come up. You have the reference genome alignment, which is just a standard check to look at. You have the issue of potential snip flips within the data. And then finally, you're able to go ahead and merge. And so I just want to highlight some of the flags here. The merging step within Plink is actually quite simple. But I guess, yeah, it can be complicated more depending on how deep you want to go in the documentation. But in this case, for our application, all we're doing is taking a Plink binary file, and this first argument is our query data set from the HapMap simulated data. And then we're invoking this B merge or binary file merge with our reference data. And so that's our 1,000 genomes data set specified here. Just all three Plink file formats. And then we have a couple extra flags, particularly this allow no sex one. Since we're only working with the autosomes, we're basically telling Plink, hey, don't worry, these are still valid genetic data. Just know that we've excluded the sex chromosomes. And then output these results into.
We're gonna go a little bit over too because we started a bit late.
Oh yeah, we're gonna go a little bit over, started a little bit late. I know that it's 2 o'clock, but we're almost fine. We're actually at the point of some nice visualization and not just looking at a ton of text. So, right out here, okay, now I see the mistake. Not that it affects our analysis. So after you merge, these files should all have the same genotypes. That should have all been taken care of, but we now have about a file data set with about 1 million variants across 739 people. That's our query data set merged with our 629 reference individuals. I did the LD pruning step previously, but I bring it back here because the pruning actually should happen at this step. Here, we're going to run the LD or linkage disequilibrium pruning function by using this indet pairwise sliding window option here that I mentioned previously. And so this is going to downsample our data set from 1 million variants down to 100,000. And then we're going to actually proceed with our PCA. And so when you run the LD pruning feature, you can see Plink will spit out for each chromosome how many variants were pruned from that chromosome and how many remain. Once you do that, here we are, finally, down to about 128,000 variants among 739 people. Finally, and I say finally, because we're actually at the step of doing the analysis. And so most of the work for anyone who's done this kind of SNP data analysis goes into not actually conducting the analysis itself, but preparing the data and making sure everything is clear. These file formats can get quite hairy, and they do require, at least in the beginning for a novice, someone who has a bit of a trained eye to help and look for certain technical issues that can arise. But I'm very grateful that you guys stuck out with me through this step, because finally we'll actually visualize some data. And so if you attended Dr. Fazari's biostats talk, she gave a brief overview of what principal components is. In this case, what we're trying to do is use principal components analysis on our merged reference and query data set to collapse the genetic variation and see which principal components essentially explain the variation that we see in our data. And I'm not going to walk through this Python, I guess, blob of code, but essentially all we're doing here is using Jupyter Notebook and Python to specify what colors to use, format the data tables that go into this, which are the outputs of Plink, as well as a metadata table that contains the population labels for each sample in our reference. And that looks something like this. And a bunch more formatting and table reorganizing, which should be not foreign to anyone that has done any sort of plotting. And boom. All right, finally, we have some results to talk about. So I'm going to zoom out a little bit and go through this a bit quickly. And so I'm zooming further in. So what I've plotted here, and this plot, actually, if you go in, you can adjust this by adjusting the group PC here. You can plot all the way out until PC 9 and 10 if you would like to, just by changing those values. But as you can see, what I plotted here are the first two, the results of our first two principal components from our PCA. And so our reference populations that I've mapped are in these four colors: blue for Africa, green for the American reference population, Asian in purple, and European in pink. And OWN refers to the HapMap individuals that we have simulated data for. And as you can see, right out the gate, compared to what Sri showed in her PCA of the 1,000 genomes data, where you see this kind of triangle shape, you still see it here, but within our samples themselves, they tightly cluster around the European group. And this was to be expected, since when generating the simulated data, we essentially just had them all come from European background to be homogeneous. But what I really want to show you guys, because that's not as visually interesting, is what happens when you actually have admixture within your cohort. And these are the data that Sri briefly mentioned from the analysis that Anthony and I conducted using SNP data from Deepa Rastogi that was generated from these pediatric obese asthma patients from here in the Bronx. So these are individuals of Latino and African-American descent that we know by being from the Bronx and from their self-reported ancestry, that these are human populations that have undergone different degrees of admixture. And so I'm not going to go through any code here. All I'm going to do is just give everyone a quick visual preview of what admixed data looks like. And once again, this should look familiar. This is the same PCA that Sri showed, just highlighting in X here, the asthma patients that we were interested in. And as you can see, they scatter actually quite vastly along this first principal component and tend to cluster all throughout, but primarily with African, but also many other population groups spread along PC1. And so if you compare that to how uniformly clustered these individuals are, here we can clearly say that these individuals, at least with respect to our reference population in the HapMap data, or in the 1,000 genomes data, that these are European individuals, or have a lot of European ancestry in their genetic background. But the association becomes less clear when we deal with, in this case, these real world patients from here in the Bronx. And so if we wanted to actually go in to the at the individual sample level and understand the proportion of their genome that comes from, said, a given population background, that's where something like admixture analysis comes in. For those that are interested in doing a bit more reading and learning about one of the ways you can do this, this second folder in the vignette has all the necessary details for that. But essentially, what we're able to do using the Plink data that I've shown you how to QC and whatnot is run admixture analysis. And I'm going to quickly go down here. So essentially, all you do for your admixture analysis, once you've done this more arduous Plink QC or gene typing quality control, you run this clustering program that essentially will estimate the amount of global ancestry proportion for each individual based on the allele frequencies that they have for the set of SNPs that you provided. And admixture will take in your Plink file and spit out two sets of results, which are these P and Q files. And these files will contain, one, the allele frequencies for each SNP inferred from each population, and then these Q files will have the actual inferred individual ancestry proportions for a given number of ancestral populations. That's K, which I'm going to speed through here. But essentially, if you want to, feel free to take a look at this code. We can use these data and plot these data to gain an enhanced resolution of the admixture that occurs within our data set. And so here, K refers to the number of ancestral populations, which is inferred and kind of given for this analysis. So in this case, we're interested in the results of this K equals 5 parameter, as we're interested for this analysis to see the global ancestry proportions within our group. And those global ancestry proportions are from the 5 sort of superpopulations that exist in the 1,000 genomes data set. And so when we actually zoom in on our asthma panel, which if I have the PCA next to it, be a bit more clear, but where we know that there's demonstrated degree of admixture, you can see just within our admixture panel, or our asthma panel itself, that there are varying degrees of admixture with varying ancestral proportions for each individual. So for example, this green bar where each individual is these are essentially thousands of bar plots that are just stacked together. And in this case, on our right, in this little right-hand corner, are the 85 asthma patients that we are interested in. And as we increase the number of ancestral populations from K equals 2 down to K equals 5, 5 representing the number of superpopulations that we're interested in, you can see different degrees of admixture and different potential ancestral backgrounds for a given patient. One that sticks out in particular, and we know that these are individuals from the Bronx of Latino and African-American descent, but in this case, you have actually someone who demonstrates quite a high degree of their ancestral proportion being from this green bar. And green is this ancestral component that we see that's highly enriched within Asian population groups, which wouldn't be expected in this case, but it is the case. And so purple here refers to a Southeast Asian ancestral component. And so you can start, you don't want to overinterpret these plots, but the way, the insight that this gives us and the corresponding output of the admixture analysis itself helps us control for varying allele frequencies that might not be as obvious when just going, say, off self-reported race or self-reported ancestry, or even just like this PCA. But yeah, this, if you compare this to, say, once again, our previous example where you have a very homogeneous population, the utility of something like this, especially for conducting studies here in the Bronx, becomes quite important. I think with that, I've gone over, but please feel free to reach out with any questions you may have when conducting this analysis. But yeah, I'll end there. Any questions? Yep.
Got one question.
Oh, yeah. Mhm. It does. So at each site, it will report the major allele, like the reference, and then it'll show you, Plink will store what that individual has. So they'll either have that reference allele, which is typically the more common allele, or Plink will store the fact that they have a variant at that site. And so, yeah, by doing that, they'll also store either heterozygous or homozygous variants that way. So this is why Plink splits this up into 3 separate file formats. I think the binary file format is where the FAM files, where I think this is actually represented as the matrix. But yeah, the heterozygosity or homozygosity of a given variant is stored at the per individual level. So each individual gets a genotype call and will be marked either heterozygous or homozygous for that variant. And Plink will also store whether that variant is the reference allele or the alternate. Yeah.
You PCA on this matrix that has information. That's like is that consider like a categorical representation? You have like you can have reference allele or everything. Are you principal ones on like category? Usually, like when you PCA, like it's like you think there's just numbers like they just put numbers. But it seems that you're not getting like actual categorical representation of the type of representation that is that right?
I think.
We could ask Dr. Rose about that.
Yeah.
And yeah, you know, so it's not really like the binary thing. It's like it will have like a weight. That's why they can separate in space, because even if they have the same variance, for me the variance is more frequent than for example. So that's why variance separate in space. So it I don't know how exactly how Plink integrates all that information to calculate the PCA, but it's not just for the
All right, considering the time, I think we should probably call the session here. So thank you all for attending and really appreciate your interest. And again, thank you to David. Please everyone give another round of applause.
Thank you guys. Feel free to use the code.